Welcome, everyone. Thank you for joining us for today's webinar, Discovering Cellular Mechanisms and Biomarkers in Anti-PD1 Non-Responders Using Ingenuity Pathway Analysis Software and OmicSoft OncoLand Datasets from QIAGEN, presented by Dr. Devendra Mistry, Senior Field Application Scientist, QIAGEN Advanced Genomics. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, please visit www.qiagen.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Mistry. I will now turn the presentation over to him.
Thank you, Xavier. So this presentation, we're going to talk about how Ingenuity Pathway Analysis software and OncoLand software can be used in combination to discover cellular mechanisms and biomarkers in an anti-PD1 non-responder dataset. Just a quick legal disclaimer that the products I'm going to show you are meant for discovery purposes as opposed to diagnostic purposes. So as I mentioned in my title slide, we want to identify mechanism of action, biomarkers, regulators, and targets from an anti-PD1 non-responder dataset. I want to clarify that this can be actually done on any dataset, public or your own. All right, so user can also do a lot of things I'm going to show you with their own dataset as well. In part one of the presentation, we are going to focus on data mining. We're going to do that by using OncoLand from OmicSoft. So in this case, I'm going to introduce you to what OmicSoft is and what OncoLand is. Then, using OncoLand software, we're going to locate an anti-PD1 non-responder dataset. And later on, we're going to study some biomarkers, such as UGT1A6 and A3. And we're going to study these biomarkers in more details by exploring curated data from TCGA and GTech. And then finally, we are going to associate these markers with patient survival. In part two, we're going to study remaining of the data set, right? So in the part one, I just mentioned 2 genes. So in part two, we're going to do data interpretation using Ingenuity Pathway Analysis. We're going to learn about what this software is and how it can be used to study mechanism of action, biomarkers, and regulators. And then we will end with a summary. So let me talk to you a little bit about this case study, right? So interaction between PD1 and PD-L1 is a very well-known immune checkpoint. And that's why there are so many drugs that are designed to block this interaction. One of the drugs is nivolumab, offered by BMS. This is also known as Opdivo. So if you watch TV, you might have even seen commercials for Opdivo. It blocks PD1. It is used in indications such as melanoma, non-small cell lung carcinoma, renal cell carcinoma, and others. Now, the problem is many PD-L1 expressing tumors, they don't respond to therapy. So our whole question is why? And as I mentioned, we want to start by maybe locating an interesting data set. So say you were to do this on your own, then typically what you would do is you would go to public domain. So you'll be going to different sources that are listed over here, such as TCGA, GEO, and so on. And then you'll be locating interesting data set or data sets, right? Now, if you want to thoroughly study this data set, then you'll be, first of all, downloading that. If it is an NGS data set, then you'll be QCing it as well. So you'll be checking, like, what is the quality of reads and such. Afterwards, you'll be processing this data set through secondary analysis pipeline. So again, if it's an NGS data set such as RNA-seq, you'll be mapping read to the reference, and you'll be QCing, like, okay, how many reads are mapped versus how many reads are not mapped. And then finally, you will do things such as quantify gene and transcript expression, and then calculate differential expression between groups of your interest, right? So you might be comparing non-responder versus responders, which is what we are going to do, or you might be comparing disease versus normal, treatment 1 versus treatment 2, right? This will depend on you. But basically, if you were to be dealing with public data set, you'll be doing all of this, right? Now imagine you're really doing the same thing with hundreds, if not thousands of public data sets, right? Because you want to generate a powerful hypothesis or you want to generate a powerful discovery. You would have to repeat this process for hundreds, if not thousands of data sets. And in addition to that, you would have to curate metadata for all of this data. So each data set will have description, right? What tissue is it coming from? What cell line is coming from? If it's coming from some sort of treatment, what was the drug concentration? What was the time for the treatment? It's patient data or is it in vitro data, right? So you'll be curating all of this metadata as well. Fortunately, our Omics team has already done all of this. So they have curated data from many different public sources that are listed on the right-hand side, such as TCGA, which stands for the Cancer Genome Atlas. This has a lot of tumor tissue information as well as normal tissue information, and normal tissue will be adjacent to the tumor. GEO, which has a lot of public data sets, same with Array Express. CCLE, which has cancer cell line information. GTEx, which has information for normal tissue, right? So our team has gone through all of these different public sources on the left-hand side and curated a lot of data and metadata. Furthermore, all of this expression data from public sources is passed to the same pipeline. So user can do cross-project and cross-collection comparison. All of this is stored in Omics Lens database, which is the image on the lower right corner. And this database is maintained and updated regularly. So let me tell you just a little bit more about this database. User can have access to this database through a couple of different products. I have listed 3 of those products over here. One is Oncoland, which has oncology data, again, from sources such as TCGA, GEO, CCLE, and others. DiseaseLand, which has non-oncology data. So this would be data relevant to cardiovascular diseases, metabolic diseases, neurological diseases, and others. And then finally, GeneticsLand, which has a lot of variant information. So this can be germline variants, this can be things like GWAS and PWAS association studies, reference collection, and more. And if you go to our website, we have greater description for all of these products. And the cool part is user can also upload their own data, have it processed through the same pipeline, and then query across user data and public data using this lens. All right. So in our case, because we are interested in anti-PD1 non-responder, we're going to focus more on Oncoland. So first question we can ask is we want to discover mechanism of action, targets, biomarker, etc. So first of all, how can we locate an interesting data set using the software? So in this case, what we are doing is we are opening up GEO Collection within the software. I have a screenshot on the lower right corner, right? And you can see this is a point-and-click software, right? So it has GUI, so you don't really need command line knowledge. But for those of you who like to work with command line, we also offer APIs. And again, what we have done is open up GEO Collection. And at this point, user can search by a variety of keywords, such as disease name, drug names, etc. Now, what we are interested in doing is we want to locate a data set where we have responders and non-responders for anti-PD1 therapy, right? So we are actually looking for patient data. So in the screenshot on my right-hand side, you see I have selected subject treatment filter. So within software, you can specify that you're interested in patient data as opposed to, say, in vitro data. So subject treatment will focus more on patient data. And over here, when you click on it, you see a screen opening up where I'm typing nivolumab. So as soon as I start typing nivolumab, some hits come up. So I'm selecting those hits and then pressing OK. So when I press OK, what the software will do is it will pull up all the patient studies relevant to nivolumab, right? And those studies are listed in the bottom table. Now, one of the great things is our team has curated metadata, so you can actually learn more about these studies by simply studying the table. But you can see there are some also links as well. So reading the links, you can read more about the studies and decide which studies you want to focus on. So in my case, I'm focusing on the study with GSE ID GSE 67501. Why this specific study? Well, in this study, authors are working with metastatic renal cell carcinoma patients. They are administering these patients with nivolumab. This study, by the way, is performed at Johns Hopkins. And afterwards, patients are classified as responders and non-responders by response evaluation criteria in solid tumor. Afterwards, they extract RNA and then use microarray and Illumina chip to perform gene expression analysis. What I have over here is just summary of methods. If you want to read about this in more details, you can, of course, follow the citation at the bottom and read about this in a lot more detail. So now that we have located an interesting study, the question is, can we look at the gene expression data from the study so we can, again, start identifying mechanism of action, biomarkers, etc. Within the software, using the GSE ID number that I emphasized on previous slide, you can easily open a couple of different views when it comes to gene expression. You can open up a table view, which I'm showing you on the right-hand side, where you can have a list of genes with differential expression in terms of log fold change, p-value, adjusted p-value, etc. Or you can also open up a volcano plot, which is in the left-hand side. And volcano plot has log fold change on x-axis and anti-log or p-value on y-axis. So using either table or volcano plot, you can study the genes that are highly up and down regulated and are significant. So in our case, we want to start discovering like biomarkers and targets, right? So what do we do? So in this case, we can study either table or volcano plot. So in the table we are doing, in the table and volcano plot, we are doing comparison of responder versus non-responders, right? So if you want to focus on genes that are upregulated and non-responders, then we'll be focusing in the upper left corner, which is what I have circled using the red circle. And when we focus on those genes, what we come across is a lot of UGT1A family members. So if you read the paper that I mentioned, they emphasize greatly on this UGT1A family members, and that's why I'm going to start discussion about them, right? So one of the members they highly emphasize on is UGT1A6. And if you read about these members, then you'll quickly realize that these members are involved in metabolic and solute transport functions. So one can even propose a hypothesis right now that, one of the ways anti-PD1 non-responders might be resisting the therapy is, these genes might be detoxifying exogenous and stress-related lipids by breaking them down and excreting them outside of the cell, right? So now we have come across an interesting mechanism of action, a gene that can potentially serve as both biomarker or a targetable molecule. So say you want to learn more about this gene. Say you want to know how this gene is expressed across different tumor tissues. And you want to also compare things like how is it expressed across tumor and normal tissue. What do? Within the software, we first open up Geo Collection, right? Now we are opening up TCGA Collection, the Cancer Genome Atlas. And as I mentioned, TCGA Collection will have information about tumor tissue as well as adjacent normal tissue. So when we search for this gene with a few clicks, we can easily open up this chart. And this chart will show you how this gene is expressed across different carcinomas. And again, I would like you guys to remember that all of the samples are processed using the same pipeline. This is something our team has done very diligently. And because of that, you can easily do comparison in terms of how this gene is expressed across different carcinomas. So in this case, you can see this gene is clearly expressed more in renal cell carcinoma compared to something like AML. Now, say you want to focus more on renal cell carcinoma. Because our team has also curated metadata and introduced filters, which you can see on the left-hand side, you can easily click on renal cell carcinoma disease filter. And when you do that, you remove all of the other diseases and you only keep renal cell carcinoma. And you can see what we see is some blue samples and green samples. So blue samples are tumor samples and green samples are adjacent normal samples. So right now they're in the same row. Say you want to split it in 2 rows. So fortunately in this software, you can also have grouping options. So you can easily split that 1 row into 2 rows where you can divide things between tumor and normal. And another thing you can also do is turn on statistics. So in the lower right corner, we have turned on statistics. So you can see at least for UGT1A6, there is some difference in the sense that it's slightly more expressed in tumor compared to normal according to this chart. And significance-wise, it's borderline significant, but there's not a great difference. Now, many investigators will tell me that tumor is not one thing, right? It has many different stages. So based on the metadata that is curated by the team, can we split this into different stages of tumor? Yes, it is very easy within the software. You can subgroup things into different stages, as you can see over here. So now tumor row is split into stage 1, 2, 3, 4, etc. And again, difference-wise, we see some difference, but the difference is not that great. I mean, there's some difference between stage 1 and 2, 2 and 3, etc., but it's not that great. Now, keep in mind, UGT1A6 is just one of the family members. What about other family members, such as UGT1A3? So we can also search for UGT1A3, and again, we see the same pattern that like UGT1A6, UGT1A3 is expressed also more in renal cell carcinoma as opposed to, say, something like AML, which is at the bottom of the screen, right? Like UGT1A6, we can split and filter, and what we see is for UGT1A3, there is greater difference compared to UGT1A6. And statistically speaking, we also see more significant p-value in the lower right corner. Finally, when we split the tumor row into different stages, we kind of see this increasing pattern as we go from stage 1 to stage 4. So I personally feel that both UGT1A3 and 6 are expressed more in renal cell carcinoma compared to other, but UGT1A3 might have better application as marker based on the charts we have seen so far. Now, the problem here is many people don't like the normal tissue that they see over here because this is adjacent normal tissue, right? So many people feel that this adjacent normal tissue may or may not be normal. So what they want to do is they want to compare tumor tissue from TCGA with normal tissue from GTEx database. So GTEx database, by the way, only has information about normal tissues, right? So they want to compare tumor tissue from TCGA and normal tissue from GTEx. And again, because our team has diligently curated all of the expression data using the same pipeline and stored that in LAN database, you can easily make this comparison. So you can open up what we call virtual LANs, right? So virtual LANs will be a mixture of different collections such as TCGA, GTEx, and so on. And using that, you can generate charts like the ones I'm showing you over here. And again, I want to emphasize this is like point and click software. If you want command line, then you have option for that as well. But if you don't have command line experience, you can easily still like point and click and generate the charts like the ones I'm showing you over here. So in this chart, what we see is we have tumor from TCGA as the top row. Solid tissue normal, which is the adjacent normal tissue from TCGA as the second row. And then finally, normal tissue from GTEx as the third row. And what you see over here is there's not much difference in expression between normal of TCGA and GTEx, but there's greater difference in expression between both normals and tumor from TCGA. So again, marker application-wise, UGT1A3 might turn out to be a good marker because there's clear difference between tumor and normal. And again, we can turn on statistics in the software. So in the lower right corner, you can see the p-value, and it is significant for this cross comparisons across 3 groups. So in summary, we found that UGT1A3 and A6 were both expressed more in renal cell carcinoma compared to other tumors, but UGT1A3 had greater difference in expression between tumor and normal tissue. And this was true both for TCGA, tumor and normal, and also TCGA versus GTEx, tumor and normal. And then UGT1A6 can potentially serve as marker, but it had lesser degree of difference. Now, one thing I do want to remind you is I have gone through only 1 or 2 collections, right? So we went through TCGA, GEO, but keep in mind, our curators have curated data from many other collections that are listed in the lower left corner. And one of these collections is CCLE, which has cancer cell line information. So say you as a user might want to investigate this in different cancer cell lines. And you will see similar conclusions in the sense that UGT1A6 seems to be expressed more in kidney cancer cell line as opposed to, say, thyroid cancer cell line, which is one of the cell lines at the bottom. So what else can we do using this software? The answer is you can actually do a lot of different things, but one of the other things that many users want to do is they want to divide the samples based on the criteria they decide. So one of these criteria can be expression criteria. So say you wanted to divide the samples between samples that have high expression of UGT1A6 and low expression of UGT1A6, then you can very easily do that in this software using what we call custom query. All right, so you can make a custom query for UGT1A6 like I have done, and you can divide samples that have high expression of UGT1A6 versus low expression of UGT1A6. And then once you divide sample based on the criteria you decide, you can generate many different types of plots like survival curves, heat map, and other plots as well. So in this case, say you wanted to test the value of UGT1A6 as prognostic marker, then you would be maybe looking at something like survival curves. And what you see is when you have high expression of UGT1A6, which is the blue line, there is relatively poor survival compared to when you have low expression of UGT1A6. Now, this is across all TCGA tumors. And if we look at the log rank test, which is in the upper right corner, then we can see this seems to be significant. And we can do the same thing across renal cell carcinoma patients, right? So remember, in this software, we have filters, right? So we can filter it for only renal cell carcinoma. And again, we see the same pattern in the sense that when you have high UGT1A6 expression, there is relatively poor survival compared to when you have low UGT1A6 expression. Though I have to say that when I turn on the statistics, this does not turn out to be significant. Nevertheless, we can say that we are seeing similar pattern. And cool thing is, you don't have to stick with one marker. You can actually check this for dual or more number of markers as well. So over here, now we have two lines. First line, the blue line, is for samples that have high expression of both UGT1A3 and UGT1A6. The second line, green line, is for samples that have low expression of UGT1A3 and A6. And you can see when you have high expression of both of these markers, A3 and A6, there is relatively poor survival compared to when you have low expression for both of these markers. And again, I want to clarify, this is across all the tumors. And if you look at the p-value, it is significant. So my summary for this would be there might be some prognostic value for this UGT1A family members in the sense that when we had high expression, we saw relatively poor survival across different tumors. And at least when we filter it down to renal cell carcinoma for UGT1A6, it does not turn out to be similar. It does not turn out to be significant, but we still see similar pattern as we would expect when you have high expression, there is poor survival compared to when you have low expression, there is slightly better survival. The bigger point I want to make is user can choose to divide data based on the criteria they like, right? So in this case, we divided samples based on samples that had high expression of UGT1A6 versus low expression of UGT1A6. In your case, you can use gene of your interest. So instead of UGT1A6, you can pick like say EGFR or LAG or whichever gene you like. And you can say, okay, divide sample based on high expression of this gene versus low expression of this gene, or divide sample based on high expression of gene 1, gene 2, and low expression of gene 1, gene 2. And you don't have to just stick with expression markers. You can also divide samples based on histological markers. So you can divide samples that are HER2 positive versus HER2 negative, triple negative breast cancer versus non-triple negative breast cancer, or go with mutation markers, samples that are mutant for TP53 versus wild type for TP53. The idea here is once you divide sample based on the criteria you like, you can generate many different types of plots that I'm showing you over here on this slide. You can generate survival curves. So we have survival curves for TP53 mutant versus wild type patients in the, UGT1A6 mutant versus wild type patients on the right-hand side. You can also generate heat maps. So we see heat map for comparing two different groups of our interest. And you can also generate things like alternative splicing view in Genome Browser, which is in the lower left corner. In addition to that, I have seen people working with RNA-seq fusion, copy number variation, mutation, viral and bacterial count using Oncoland software. So now that we have gone through data mining, now that we located an interesting anti-PD1 non-responder data set, we also studied biomarkers such as UGT1, A3, and A6 across TCGA and GTACs and associated them with survival. Let's do interpretation. So far we have studied just two markers. There were also other genes that were significantly up and down regulated. So now we can ask questions, are they connected with mechanism of action? Are they connected to potential biomarkers? Are they can some of them be, interesting regulators and targets? So IPA is a software where people typically start with a table of molecules of their interest. So these molecules can be genes, right? So in our case, we had this comparisons between non-responder and responders, and we had list of genes that were, differentially regulated. So you can start with a list of genes, list of transcripts, list of proteins, list of metabolites, list of microRNA. And idea here is you have a list, but you don't know, what different molecules in that list are doing, what different genes and proteins in that list are doing. So that's where Ingenuity Pathway Analysis and its database come into play. So using this database, Ingenuity Pathway Analysis is going to convert this list into different visuals that are on the right-hand side. So now instead of visualizing lists, you can see what pathways this list is connected with, what biological processes this list is connected with. You can also open up different pathways and processes, and you can see how different gene expression is impacting different parts of the pathway. So you can study about mechanism of action. You can also study biomarkers through creating bar charts and heat maps that I'm showing you on the right-hand side. And you can also compare mechanism of action using the heat map. So let me go into all of this in more details. The key here is user has a list. This can be differential gene expression, protein list, metabolite list, and user is confused, like, okay, what is this list related to? So that's where IPA comes into play, and it will allow you to do interpretation that I'm showing you on the right-hand side. So one of the reason IPA allows you to do this is it is backed by this powerful database. This is what we call Knowledgebase. In this Knowledgebase, we have curation of peer review publication. A lot of this peer review publication is manually curated by PhD level scientists, and this is updated on weekly basis. So basically, we have curation of high quality information, and this is updated as frequently as weekly basis. In addition to that, we also have curation from third-party data sources. So this will be data sources such as BioGrade, which has protein-protein interaction, clinicaltrials.gov, which has information about clinical trials, Tarbase, which has information about microRNA, HumanSick, which has information about metabolite and pathways and such, right? So we curate information, our team curates information both from peer review publication and third-party data sources. And one of the great things is this information curation is quite contextual. So say you upload a list of genes or you upload 2 genes, then IPA won't just show you that gene A is connected to gene B. It will give you the context that this connection is actually protein-protein interaction. This was discovered in human in such and such cell line. This was discovered by co-immunoprecipitation experiment. And most importantly, it will list the source. This information is coming from this specific publication, and if you click on the hyperlink next to the publication, it will also take you to that publication, and it will show you the PubMed page for that publication, so you can dig into it in more detail and see if you want to trust that information or not. But key here is we have a database where our curators are reading through text and figures of the paper and manually curating a lot of the information, and that's why it is of such great quality. Now, just to contrast this with the other database that I mentioned earlier, Oncoland, right? So in this database, our curators are going through text and figures and curating knowledge, as I'm showing you on the right-hand side. In the other database, Oncoland database, our curators are curating actual NGS data, which user can manipulate and generate different charts that I showed you, all right? So now moving forward with IPA, before I start showing you some charts and figures and such, it's important to know some of the legends that I'm listing over here. So in IPA, you're going to see many different colors. I request you to remember 4 of them: red, green, orange, blue. Red and green are from your data set. So red are the genes that are upregulated in your data set. Green are the genes that are downregulated in your data set. And orange and blue are the predictions IPA is making based on your data set and information that is curated from previous literature. So for example, MST1 is colored red. That means it's upregulated in your data set. So upregulation of MST1 is leading to increased apoptosis. IPA is making this prediction based on the information that is curated from previous literature. Similarly, BAD is colored green. So downregulation of BAD seems to be leading to decreased apoptosis. So again, BAD is downregulated in your data set, and IPA is predicting, according to previous literature, it will lead to reduced apoptosis. Similarly, TGF-alpha is color red. That means it's up in your data set, and this is going to lead to increased activation of ERBB2, also known as HER2. So now that we understand some of the basic legends, say you were to upload non-responder versus responder comparison in IPA, so differential gene expression for non-responder versus responders. So IPA will give you a list of pathways that I'm showing you over here. And then if you go through these pathways, you will quickly realize that these pathways are cancer-related or are related to growth, growth factor, inflammation, and cell cycle. All of this are cancer-related as well. So basically, the top pathways we see in IPA, they're all cancer-related for this anti-PD1 non-responder data set. And you can open up pathways. So in this case, to study mechanism of action, we are opening up molecular mechanism of cancer pathway. And you can see pathways are nicely organized from plasma membrane to nucleus. In this case, we have plasma membrane both on top and bottom, and we are going to nucleus in the center. If we zoom in the pathway, then we can see the functional impact of gene expression on this pathway. So for example, in the center of the screen, you have NBS1 color red. So red means it's upregulated in your data set. And this seems to lead to increased DNA repair because DNA repair is color orange. Similarly, if you focus in the lower right corner, it seems that differential gene expression is leading to increased cell cycle and reduced apoptosis. So now what we are figuring out is differential gene expression that is present in non-responder versus responder is leading to increased DNA repair, reduced apoptosis, and increased cell cycle. And this explains why non-responder might be resisting the therapy. Similarly, if we look at the bottom part of the pathway, we see LAF1 red. Red is upregulated. If you know about LAF1 is a cell survival marker, right? If you did not know about it, you can easily find that out using IPS database. And what you can see that upregulation of LAF1 is also leading to increased cell survival in anti-PD1 non-responders. So again, this is another way they might be resisting the therapy. Now that we have talked about mechanism of action, what about biomarkers? What type of biomarkers can you discover in IPA? I already mentioned one biomarker, which is LAF1, but in IPA, you can turn on this bar chart, and this bar chart will show you how LAF1 is up in non-responder, which is the red bar, and down in responder, which is the green bar, right? So again, we would expect cell survival marker LAF to be up in non-responder and down in responders. And then similarly, other markers, such as the ones you see on the right-hand side, such as CDKN2C, BERK3, they're also all up in non-responder and down in responder. What if you did not know what this marker were? Then you can easily use Ingenity's database and figure out what these markers are, which is what I have done over here. So you can see markers such as CCND2, NBN, LAF1, they're all cell viability markers, and they're up in non-responder and down in responder. Similarly, other markers that are related to, say, cell proliferation, such as PSEN2, MAP kinase 30, they're also up in non-responder and down in responder. And then finally, you're like, okay, you're telling me that these markers are cell viability markers, these markers are cell proliferation markers. Based on what? Then again, in IPA, you can turn on this relationship view page, which will tell you where this information is coming from. So for example, CCND2 information is actually coming from cell cultures, and it is coming from MKN45 cells, as I'm showing you at the bottom. The authors have done siRNA library screening, and you can actually go to the specific publication listed at the bottom by clicking on the hyperlink and check out this in more details if you want to see how they derive this information. So you can always verify information. When it comes to biomarkers, many users actually want to find biomarkers based on other criteria. So some users, for example, they want to find biomarkers based on their subcellular location. So say you're looking for biomarkers in membrane. Then we also have tools such as Bioprofiler, which will allow you to use various filters and focus on biomarkers of your choice. And over here, we are discovering that B2M can be one of the membrane biomarker that you can focus on in anti-PD1 non-responders, as I'm showing you over here. Now that we have focused both on mechanism of action and biomarkers, what about regulators and targets? So in IPA, we have some powerful tools such as upstream analysis and causal networks, which will allow you to focus on regulators and targets. Using that, I have discovered over here many transcription regulators that are connected to renal cell carcinoma. and also found to be activated by IPA. And you can see them in the center of your screen. And if you're curious what these regulators are, a lot of these regulators are, again, involved in processes such as cell cycle, DNA repair, and such. And similarly, if you focus on genes such as GFPT1 and SMITE3, these are enzymes. So if a user is looking for targetable molecules, now you have targetable molecules. So IPA can easily give you master regulators, and it can also provide you with some interesting targets. And say you wanted to study this target in more detail. So say you wanted to study GFPT1 in more detail. You can actually open up the network view, and you can easily see how GFPT1 is connected to renal cell carcinoma. So it seems that GFPT1 is activating TGF-beta1, MYC, and TP53, and this seems to be leading to increased renal cell carcinoma phenotype. So while we were looking for interesting target, we can also actually dive into mechanism of action, and we can see how activation of GFPT1 is leading to activation of downstream regulators, and how that is leading to increased renal cell carcinoma phenotype in anti-PD1 non-responders. And then finally, for those of you who might be interested in drugs targeting different genes on this network, in IPA, you can easily figure out, like, which drug can target which gene. So over here, I have a drug targeting TGF-beta1, and you can find out additional information about drugs, like is this drug an agonist, antagonist, what clinical trials is this drug involved in. So in the table, you can see this drug is involved in different phase 1, phase 2 clinical trials. What is it? It's an immunomodulator. What does it bind to? It binds to different members of TGF-beta1. So over here, I have just turned on one drug, but honestly, you can turn on drugs for many of the factors that you see on this network over here, right? So again, to summarize, we have looked at like one of the mechanism of action example. We have looked at some biomarker example, and we have also looked at regulator example. But so far, we've been studying one thing at a time, right? So we've been studying like one mechanism of action at a time using a pathway. We've been studying one biomarker at a time, and we've been studying like one, regulator at a time when we look at the network. What if you wanted to do comparison across multiple networks, multiple regulators? In IPA, you can turn on this heat maps, and using this heat maps, you can compare multiple groups, right? So over here, what I have told IPA is show me only cancer pathways and show me how they are differentially activated between non-responder and responder data set. So in one column, I have non-responder, which is the orange column, and other column, I have responder, which is the blue column. And orange and blue, again, are predictions made by IPA. And you can see a lot of these cancer pathways are on in non-responders and off in responders. So for those of you, again, who are looking for mechanism of action, you can kind of see how different cancer-related pathways are on in non-responders. And this can be one of the ways non-responders are resisting the therapy. Furthermore, if we dive deeper into the pathway, we can see the specific, like, for example, if we dive in IL-7 signaling pathway, then we can kind of see CCND3 is upregulated, and that is leading to proliferation of cells in non-responder. Similarly, other genes up and down regulation seems to be leading to reduced apoptosis and altered metabolism in terms of like increased glucose uptake. So in terms of mechanism of action and generating hypothesis, in addition to looking at the heat map, you can also turn on networks, and these networks will give you greater insight. So let's take it a step further. So far we've been comparing only two groups from the data set that we are studying, which is the anti-PD1 non-responder data set. But there are other data sets that are out there in TCGA and GEO that have to do with renal cell carcinoma. So now the question is, can we compare the data set we are studying with public data set from TCGA and GEO and see if the mechanism of action is similar or different across this different renal cell carcinoma data set? And another thing we can also ask is, can disruption of different protein lead to similar phenotype when we do this comparison? So as I mentioned earlier on when I was covering Oncoland from Omicsoft, is in LAND, our curators have reprocessed all of the expression data using the same pipeline. And what our IPA team has done is they have uploaded many of this comparison in IPA and then run core analysis on it. And now every single time you upload any data set or any comparison in IPA, And by comparison, I'm talking about like non-responder versus responder comparison. Anytime you upload such comparison, your comparison is automatically compared to other comparisons. And this other comparison can be your own comparison, or they can be also public comparison. So again, in simpler words, your IPA analysis is compared with your other IPA analysis. So if you have treatment 1, it will be compared to your treatment 2, 3, 4. And it is also compared to public analysis of different data sets. So in this case, we are going to, again, compare anti-PD1 non-responder data set with public renal cell carcinoma data sets. So for this, we have a heat map. And in this heat map, the left-handmost column is non-responder versus responder comparison, which we have been focusing on so far. And it is being compared to different renal cell carcinoma data set from public domain. And you can see we actually come up with some very interesting comparison in the sense that some of the data sets on the right-hand side that are in the bigger square, these are the data set where authors are comparing disease renal cell carcinoma tissue versus normal renal cell carcinoma tissue. And you can see the mechanism of action we see in our renal cell carcinoma data set is similar to what we see in this other public renal cell carcinoma data set. And I don't have it on this slide, but the second to last data set is actually a very interesting data set where they use humanized mouse model. And, you know, that like shows similar behavior as like human data set. And then in the center, you know, I have also circled like 2 columns. And at the bottom, I have labeled them BERK3 and EPAS1. So BERK3 is a protein that's involved in processes such as apoptosis, invasion, cell cycle, and so on. And EPAS1 has been shown to be one of the anti-tumor proteins in the past. And idea here is the comparison you're seeing in the center is mutant versus wild type for these genes. So you can imagine if you were to produce mutation in a gene that is involved in apoptosis, cell cycle, or in tumor suppression, you might get a profile that is more similar to tumor, and that's exactly what we see in the center, right? So my bigger conclusion on this slide would be that mechanism of action that we see on in non-responder is also on in other renal cell carcinoma data set from public domain. And then in terms of which data set is coming from which study, you can easily click on the title and you can find out more about the study by looking at the table within the software. So this table will give you a link to the study, so you can go on that link and read more about it. But in addition, it will give you information such as if the authors are using microarray or RNC, or what type of tissue they're using, what type of cell line they're using, what exactly are they comparing, what type of statistical test they're doing. All of this is easily visible within the software by simply clicking on the title. Now, using Heatmap, if you wanted to study mechanism of action in more detail, so for example, if you come across interesting regulators such as CD44, OSM, and you wanted to know how these regulators are connected to tumorigenesis of tissue, you can easily click on the squares next to them, and you can open up that network, and then later on you can connect that network to tumorigenesis of tissue, which I have done on the right-hand side. So now you're not just saying that mechanism of action is similar, we can say the mechanism of action that is leading to tumorigenesis of tissue is similar between anti-PD1 non-responder and other renal cell carcinoma data sets. And then finally, I want to make a distinction over here. Many times people confuse this heat map with gene expression heat map. So keep in mind, the heat map that I have on the left-hand side, which has orange color, this is not a gene expression heat map. In this heat map, what IPA is doing is based on your gene expression data set, it is determining what factors and what pathways should be on or off, right? So it determined that OSM should be on, and it has colored it orange. But now you're wondering like why OSM is turned on. You can also generate another heat map in IPA, which I'm showing you on the right-hand side. So the gene heat map on the right-hand side is the gene expression heat map. And this gene expression heat map shows why OSM is on, because many of the downstream factors of OSM are also upregulated, and that's why OSM is on. So this is a very important distinction. The heat map on the left-hand side is activity heat map. The heat map on the right-hand side is gene expression heat map. And by looking at gene expression heat map, we can also discover other biomarkers that are on not only in non-responder, but also in many of the public data sets that we see on the right-hand side. So in summary, what we did is we learned the mechanism of action by looking at both pathways and networks. So if you remember, we looked at molecular mechanism of cancer pathway, and we learned how differential gene expression was leading to increased DNA repair, reduced apoptosis, and increased cell cycle. And we also looked at interesting networks such as GFPT1 network, where we saw increase in TGF1 activity, also increase in TP53 activity, and how that was leading to increased renal cell carcinoma phenotype. In addition to that, we also studied some biomarkers such as LEF1, CCND2, and others, which are showed both as bar chart in the lower left corner of the screen and a gene expression heat map. And then finally, we also compared mechanism of action between data set of our interest, which is the non-responder data set, with public data set. And this is the heat map that has orange color at the bottom, right? Now, what I did not go over is the scenario where user does not have data set. So keeping this study in mind, say you're a user, you don't know anything about UGT1A6, which is what we studied earlier on. And you're like, okay, how is UGT1A6 connected to renal cell carcinoma? Even without data set, you can generate the network I'm showing you on the right-hand side, where you just simply search for UGT1A6 in the search bar, grow a network out of it, and connect it to renal cell carcinoma. And this is how you can easily figure out that UGT1A6 can interact with other family members such as UGT1A3, A7, as well as other genes such as androgen receptor. And this is how it can make connection to renal cell carcinoma, right? So idea here is the software I'm showing you, you don't need to have a data set to use them. Even without data set, you can generate network, and using those networks, you can generate hypotheses, design downstream experiments, and so on. So my overall summary would be, we looked at 2 different software and 2 different databases. So one software was Oncoland from Omicsoft, and other software was Ingenuity Pathway Analysis. To contrast the databases backing these 2 software, Oncoland's database has curation of actual NGS data, which is reprocessed using the same pipeline. And user can manipulate this data to generate expression charts, volcano plots, survival curve, heat map that I'm showing you in the upper right-hand corner. As opposed to that, Ingenuity Pathway Analysis has a different type of database. Ingenuity Pathways Analysis database, curators are reading through text and figures of journal articles and curating information, right? So it might have NGS information, but then in addition to that, it would have information about like Western blots and so on. So in Ingenity's knowledge base, people are curating information from text and figures. As opposed to that, in Oncoland's knowledge base, we are curating actual NGS data. And that's why these 2 databases, they are so complementary to each other, and then can be used in combination to do many different types of discoveries. So I already mentioned discoveries by Oncoland database. Now for Ingenuity database, we did discovery in terms of, we looked into the mechanism of action, we looked at biomarkers, we also compared our analysis with public analysis. So using these 2 database and software in combination, you can actually do a variety of discoveries. And I have only outlined some of them in my presentation today. So with that, I want to quickly thank my colleagues who have helped me generate this presentation. And if you guys have any questions, I'm happy to take them right now.
Thank you, Dr. Mistry, for that informative presentation. I would also like to thank QIAGEN for making today's educational webcast possible. And before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. And as a final reminder, our speaker will follow up with any questions you submitted via email. And that's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.